Okay, part two. So we went over most people's questions on the practice exam. We can go over any questions you had on the homework or any questions on the discussion worksheet.
So, the first one was homework, is that DNA is run in a native cell. Lane one represents DNA extracted from cells at time equals 0. Which lane 2, 3, or 4 represents DNA from a population of cells that consider one full rounded representation. So the correct answer was the unlimited plot first.
Okay, so the interesting thing about the problem of that gel is that it's showing you the gel from like left to right.
Yeah.
Which is what's a little bit different. And then,
So it's a 4-1 mix. That's what I was. Okay.
okay, so it's native, right? So if it's native, after one round of replication, how many bands are you gonna have total?
One.
One mixed band. So the fact that it's one band, that's why it's the answer.
Got
And
it.
if you can assume that.
And then I think the previous one said that one is where it's really heavy, right? So it's not that one, it's 4-1. Okay, so I get that answer is mixed, but would you maybe be more specific of labels on the exam or
Oh,
not?
on the exam, it's gonna be hella quick.
Oh.
So the idea is that we push the difficulty a little bit on the homework and the worksheets, that when you get to the exam, you feel familiar with the concepts. Yes.
What is it? So when you get down, like, I have to really, like, when you do the process.
Okay, so lane 4 was kind of like right here, yeah?
Yeah.
Lane 1 was kind of over here.
Yeah.
Okay, lane 2,
Next was like, kind of like a line under.
like light, a little bit here, and
That
then
one is- and then next to the line.
like this one's lighter,
Yeah.
and this one's darker.
Yeah.
And then what about this one?
Bottom is opposite.
Okay, darker and then lighter.
And these are
Oh, okay, I'll move it up there. Okay, so this is 1, 2, 3, and 4. Yeah. Yeah, and then so, I'm so sorry, T equals 0 is left, correct? Or right, what does it say? Okay, so this is T equals 0. Yeah, it's probably 15, and then as the divide is going to go this direction towards M14 to get more and more. So we're just gonna think it out. This is basically like the very beginning of the experiment, right? Everything is one band and 15. As it divides, you start getting more towards the N15, but because we're seeing 2 bands, we're gonna assume that this would be an alkaline gel or a denaturing, as opposed to something that could have only one band. So each band, the interpretation is going to depend on the condition that the gel is run in, and then how many generations you say it occurred. And then so usually when I show these on the exam, I like to give you a really clean, like heavy light, or ask about heavy light, so there's a dichotomy. It makes it very, very easy to understand. Experimentally, when people are actually doing this, you see these smears, and you're trying to figure out how many rounds of replication are occurring over time. It's very difficult to be like, I'm gonna do an experiment, and it's gonna multiply or replicate exactly one time. Because usually not what happens. It's usually you do a time series to see how this changes over time. Yes, all the questions. Clear.
So in order, by the time it's more fun.
No, for this figure, the time is not in order.
But you don't know if like next 3 was.
You only know that lane 1 is time or T equals 0. This allows you to understand what direction the experiment should go. So that way, you know which side is heavy, which side is light, and that's all this is telling you. Which side is heavy, which side is light. Mhm. Yeah, we're good. We're good. So once again, so all depends on the scenario and knowing which side is the heavy and which side is the light, and then whatever information you give the questions will help you interpret. Okay, you see right there. It can represent alkaline gel if it was dispersive.
If it asks like for alcohol, it's like.
Yeah, so what I would do, no matter what the question is, even if it seems straightforward, take your two different colored markers, just draw out what it's asking you and then match it to your results. Yes. So I cannot explain why, what I can do tell you is that it is given to you. That's gonna be the very beginning of the experiment. So for this particular figure. So remember when I said, think about the figure that is placed in front of you. The figures can be different ways, they can change. I could put a figure and say t equals 0 is on the left. So whatever's given to you as the figure legend about the information you have, use that information. So in this particular case, it says t equals 0 is lane 1. And what do we know about t equals 0 for this type of experiment? That's going to be your heavy or your parent strand. And then that's the information you go off of moving forward. You have another question?
Yeah, just about like, it says here that like the data structure is not like radius and the same experiment, one field that radius to 4 the radius, that transition from 1 to 4, going from heavy
Heavy.
all the way
Yeah.
to next. That's kind of what we're seeing in the 2 and 3 thing that.
That shows transition. Yeah.
Okay.
Yes.
For the last question about this, the party marriage is very significant.
So what's the question again? Okay, so what it's asking is basically what it's asking you, the DNA, basically chopped up into pieces, is the way to say it. So you're having multiple bands for being chopped up. So once again, on the exam, questions would be a lot more straightforward and clear. This is the test for conceptual thinking and application.
It was very basic.
Yeah.
It's not out because the first generation hasn't been here.
Over here.
That would be the next, right?
Well, this would be the mix, but so what I don't want you to do is try to, I guess what each of these would be outside of the context of what the question is asking. Every single question is going to be context dependent. And then so you have your core concept, and then you want to apply the core concept to the actual question stem that's being asked about that figure. So you easily have the same figure, have multiple questions that all ask you different things. So don't assume from like, okay, I know from my homework problem that this was dispersive or that this was native, so in this question it has to be native. Don't do that. Does that make sense?
Yeah. With, like, it's gonna be out on, it wasn't having the body. There's only like more for you, and we never got to the level side.
So on the question you said this is native, right? What the question said? The question was is native? Okay, so the question stem says this is native. So don't think about what would happen if it's alpha. You're like, this is native, this is what occurs. So really focus on what does the question give, like what is this like the MCU of this question? Question.
So technically, in that way of voice, the empathy student, you still have like one, I guess, like 7 DNA that is the original heavy empathy. It's just that it's so light in comparison to the other one. That's all it is.
So what's happening here is that this is actually one full round of what's called replication. Yes? And then so as you're going through one full round of replication, you have your parent strands separating and then replicating. As you're replicating, you're adding more and more of the light band. And then when it's all thrown together, then you have everything with one light and heavy and it's mixed. So in 2 and 3, you're seeing the transition of some of the parent strand is not actually replicated yet. So it hasn't added its partner like light strand. And that's why it's getting lighter to go across until it's all one band. So that's what you see in this particular experiment. It's a transition. Transition. Okay, yes. Yep. Oh, lose my voice. Okay. Any other questions on this particular figure in the homework? Like you can do this, you can do anything. Okay. Yes.
Like part of the
Yes.
I thought that, like, the R time, like, my, first I was like, I feel, but you go out, you kind of, is.
That's also true.
Yeah.
That's secondary.
Yeah. So,
So the thing about, this is not the exercise, DNA polymerase, you have to have that 3' OH group in order for it to add any more nucleotides. But RNA polymerase can take a nucleotide, and be like, wow, put it together. It doesn't need to have that one piece of the strand already there in order to initiate synthesis. So that's why DNA needs RNA to put the primer down first, so it has something to initiate the synthesis off of. And RNA does not need that. Is that clear? Yes.
maybe if you, really practice the, in the experiment, this one, like, her, the experiments. I mean, when you do the process,
Okay, so we talked about this a little bit in part 1, review. So what's interesting about how all these different histone proteins interact is that none of these interactions are covalent. And that's the part that you need to understand to be clear. They're not covalent interactions. They just kind of hang out with each other. They make little clicks. And then so when you run them on the gel, all the pieces will dissociate because they're not covalently linked. So even though they form these heterotetramers, they can be taken apart very, very easily on a gel. So if you don't cross-link them, then they won't run together as a unit. Is that clear? Okay. Other homework questions in general, anything? Yeah.
Yeah, are they polymerases on the exam?
Nope, you will not. You will not be expert RNA polymerase on the exam. Okay, other questions? Yes.
There's a question where it says that we have then, why are we studying that we have
What's it mutated to? What does it say? Let's give us everyone more information.
Like H instead of H,
Okay, can you do the very, very first half slowly, the part where the mutation is, so you can draw it out. Mhm. Five, is that a 5? Okay, it's 5. Okay. So we have a mutant DNTP, and at the 3' we have an H instead of an OH. What is this position normally used for in DNA elongation or DNA synthesis? So what does nucleophilic attack? The O. If the O is not there, what's going to happen? No attack is going to happen. So we get rid of that O, there's no nucleophilic attack, you cannot do DNA extension. Is that clear? This position and this OH is very, very important for DNA extension. That's the whole mechanism of how it works. Okay.
I would say that it's not being incorporated into a DNA strand, but the answer is that it can be incorporated into a DNA strand, but and not form phosphodies or balance it in the DNA.
What's on the other side of this?
But
So-
it would stop there.
Mhm.
Okay.
Okay, there we go. Yes. So it can come in and get added on because it has this, the triphosphate, right? And then this will be incorporated into the strand. Then when you try to add the next one, you can't get nucleophilic attacks and no more is gonna happen. Mhm. So remember this is the mutant triphosphate, this is what it looked like. And then you had a O over here. Nucleophilic attack, pow, pow, pow. You got your 2 inorganic phosphate or pyrophosphate, then you had the pyrophosphatase, you got PI plus PI, then now try to add this next one over here. And this is phosphate. And then this can't happen, because there's no O. Any other questions? Here, I'll just wait on my questions. Okay, so there. Any other questions? Okay, so that's good. Questions are starting to like trickle down. So in your studying, please go over the practice exam questions, how you think about them, the concepts that, if anything, if you can even arrange like your notes in order of like, these are the core concepts. So you can go back to those core concepts and revisit them. Like knowing the core concepts is what's gonna get you through the exam. Like when I say my dream is that everybody gets an A on this exam, like I need it. I 100% need. Like I think all of you have the potential to do really well on this exam. The questions you're asking, you're thinking about the problems the way I want you to think. So like I said, don't spend time on the reading. You can go back to the book if you just need a clarification on something, but the reading's not what's gonna help you. What's gonna help you is practicing and applying. So the whole way, remember, like 30 minutes ago, some of you started coming up with your own hypotheticals. Like, well, what if this was pH? It's exactly what I want you to do. Ask yourselves the what ifs. Okay, we had a lot of nucleus, but there's a little bit of nucleus. What would the outcome be? And then that shows mastery of the concept, and that's what you're going to get tested on for these. And so I'm happy to say I blocked the room till we have a whole nother 30 minutes. So yes. Oh, number 6. I know exactly what you're talking about. This was on the exam last year. And everyone hated it. So I put on discussion worksheets, so you can go over the concepts. But essentially, we're looking at double-stranded DNA. We had the melting temperature, we had single-stranded DNA. I had a couple of options basically asking how much of what things or items were true. I think for the most part, everyone was able to get these answers and not really struggle. But I think one of the options had some people like, well, how do you not know? Or how does it not change? Or like, you know what I mean? Yeah. So.
So, what we got with you was that if we look solely on the graph, the TN is constant, but the only reason this is false is because we know that we can given different circumstances, but not a given.
So in the question stem, what does the question stem say at the very top?
Based on the graph below, we do the following statement.
Based on the graph below. Remember to answer your questions based off of what is in front of you. Based off of this graph below, this piece of DNA has a defined melting temperature. Yes? Of course, you know from the class that there's ways to like change melting temperature of DNA by doing different things to it. But did we do any of those things to this DNA? And did I say we did any of those things? No. Don't let yourself get tripped up by overthinking the question. So yes, I heard a noise. So this is not true. The melting temperature, double-stranded DNA. Wait, yes, the double, no. The double, oh my gosh, that's true. The melting temperature of double-stranded DNA doesn't change.
Okay, so.
So it said it was false that the melting temperature of double-stranded DNA.
Wait, it's not a constant graph. It's changing.
Okay. The question is false. This is the actual answer that's there, right? The melting temperature, double-stranded DNA doesn't change.
Yeah.
Okay, but it's also part of the reason why I put it in discussion and not in the exam, because it was confusing. I actually gave points for both answers last year. So, but what I want you to do is think about what's in front of you. And I'm going to reiterate one more time. My goal is for you to learn and show mastery of the concept. When you bring about a question like this and say, hey, what about this? Doesn't make sense for this XYZ reason. Does that show me mastery of the concept? Yes, it does. Does that mean you get points? Yes, it does. So when you come across something like that and you are like sure off your mom, I'm like, I studied, this is what makes sense. Put your answer, we'll go over the answers, you'll tell me back, and I will give you the points because what I want for you is to learn. That makes sense? So don't let stuff like this stress you out. And I probably won't use this answer for next year. Okay, yes, question here. Yeah.
On the one where you still being able to is. The one that's in there, the range of model that's so the right answer is a smaller, you get a smaller, so you don't have to possibly there, but I thought like. If the amount of data that allows around it just down to the bases and that's then everything that you can do, but
So what is being asked is if the amount of DNA that's gonna get cut up is gonna be independent of how the nucleosomes are ordered. So we're gonna do one example here. A nucleosome was wrapped around into 150 base pairs like that. And then we said that normally it's evenly spaced out. Small amounts of nuclease produce these little cuts that in these susceptible areas will give you those 200 kind of bits. The 200 bits are dependent off of the small amount of linker DNA in between. If these weren't evenly spaced out, we could have a nucleosome all the way over here, and it could be long, and one all the way over here, and this could be long. And the whole idea is that with the experiment, they use small amounts of nuclease. So sometimes you could cut here, sometimes maybe here, sometimes we only cut here, need a long one, which is why we would get the smears. Because what's wrapped around the histone is the 150 base pairs. So instead of being able to get neatly or neat sets of about 200 or 400, we're instead getting random sets because the nucleus has there's a very small amount of nuclease. Yes.
So even if we had a lot of nuclease, but there was still histone intact, it would chop off the DNA on the system.
Oh, battery's low. Sorry. Say the question one more time.
Okay, so just like if we had a lot of nuclease, but like we didn't digest that histone to go for protease, but just put a lot of nuclease, those DNA wrapped on the histone will just be there, so you only get a band of 150.
Yes. Okay. Okay. Crisis averted. We're good. Okay, next question. Or okay, let's answer the question first before I move. No. That would be great. I mean, oh, I'm so excited. I mean, no stress. I'm excited. Here.
So the bees is, base pairs and then the string would be different.
So the base is 150, and then there's about 25 on each side if I was going to cut it. First, maybe, sir. And then the 150, 25, and 25 is how you get the units of 200. Yes. There's multiple ways you can do it here, but if you did proteinase first, you got rid of all the histones, so there's no protection. And then you did a small amount of nuclease, you probably get smears. If you did, if you had DNA and it wasn't ordered like this, and you did small amounts of nuclease, you probably still get a smear. So there's multiple scenarios where you get a smear on the gel. And then what we know to be true is that we get the pattern because of the amount of DNA that restaurant histone is protected. Yep, the small amount of nucleus is used first, you're gonna get 200 pairs. Unless it's a hypothetical. Then we'll think about hypotheticals that you think I might do. Well, Mr. Brain, I know it's early in the morning. It's not early anymore. It's like freaking 9:30 now. It's late. Or I'm some morning person or all day person. I never sleep. I'm like, like a vampire or something. Okay. Any ideas on the type of hypothetical I can give you? Yeah. Oh, yeah, that's pretty good. That's a good one. Mine too, this year. So basically saying that the interaction between the DNA and the histone is very, very low, so it's not tightly associated. And then you have small amounts of DNA. What do you think could happen? So we maybe lose some of the protection, right? Because it's not associated with the histone the way it should be. So just thinking about these different core concepts and how they impact outcome of the experiment. Say for example, nucleosomes were actually 400, compared to 400 base pairs of DNA for each nucleosome. Like, what would that look like here? You still get your 150 wrapping around, but you needed some more linker DNA, right? And you have these repeating units of 400 as opposed to repeating units of 200. Yeah. So it's all hypothetical, but you know that the units have to add to 400, correct?
So like, one thing you have 150 and then 250 of experience. 250 repairs.
I mean, you could,
Like you have.
but it's hypothetical, you said. You just know the units are 400, right? So the idea that, well, what would the result look like if your units weren't 200 or 400, 300? You just understand that the repeating units, each repeating unit represents a unit. Yes.
What would you say that there are only one guy? Is that none of the?
That's not really possible. So to get one band, you have to have a lot of nuclease and everything protected. But the idea is that if you had a whole bunch of nuclease, even these little ends will probably get chopped and you only protect what's right around the nucleus. So if you're going to get one band, it's probably gonna be 150.
So you will only have one band at the.
Yes.
I actually have a question on the discussion of the douchebag percentages, where in the example I have, we only have 5%, and then we would be answering the this species, or whatever the same with this. Like each thing.
So same base percentages, it's gonna be the same species. So same species have overall same percent of base pairs. Okay, and you had to follow up to, someone had to follow up to this? So 200 repeating units. The 200 repeating units is what we normally see from the outcome of this experiment, and it's how we learned that nucleosome consists of 150 base pairs plus the small amounts of linker DNA. But if we used ample amounts of DNA, ample amounts of nuclease, a lot of nuclease, and cut up everything, then we probably end up cutting up some of the linker DNA, then you would get one band, because you cut everything. There's no like two pieces together, cut everything. You only have that one band of 150 base pairs after you did the proteinase already. The small tiny at the bottom is when you take proteinase first, there's no nuclease, so all the DNA is naked. Everybody's exposed. So the linker DNA, what's the question? Because the linker DNA will get cut too. So it's a lot of nucleus. Okay, any other questions? It'll be repeating units of 200. There's no way to effectively cut every single nucleosome into one unit without putting in danger the extra linker DNA. Yeah, you still get about 200 at moderate. But it's very difficult to titrate a nuclease to not cut off the ends and to get only 200. So I would never try to ask you a scenario, is it going to be only 200? Because it's so. Okay, yes. Oh, this is important. And you have a question about this on the exam. So I'm glad you brought it up. Steps of percutase. We have virus, or bacteriophage. It's a bacteria virus. And then we do two things. What do we do with these viruses or bacteriophages? Okay, Marking the protein with radiolabeled sulfur, marking the nucleic acid with radiolabeled phosphorus. What's the next step? Add into a bacteria. What do they do when they're on the bacteria? Inject the material. Boop, boop. Okay, and then what happens next? You do the blender. What does the blender do? Take the phage off of the bacteria. What happens next? We centrifuge them. And then we have pellets and supernatant. What's in the pellet? Bacteria. What's in the supernatant? Bacteriophage or virus. Where do you expect to find radiolabel? The pellet. Where do you expect to find radiolabel over here? And the supernatant. So sideways. Okay, so then what would happen if we didn't do this blending step that separates the bacteria from bacteriophage? Yep, be stuck together. You centrifuge, they'll both go to the pellet. They both go to the pellet. So if they both went to the pellet, because we didn't blend, where are we gonna find signal? Both of them would be in the pellet. So is that clear? Does that make sense? Okay, good. Other questions? Yeah.
You said like, you might take a look at spec, you think that it would throw off, maybe you want the back to really take it, and then we get a little bit into that or not.
It does, really perfectly, it does. So they had to do that little white blending step and go, get off, viruses, get off of that, and then that's what they did. Okay, yes. Yes. Centrifugation just pulls the heavier thing down to the bottom of the pellet, everything else is stuck in the liquid. Or left behind the liquid. Okay, other questions? Concerns, comments? Yes. Well, better bring it. Okay, so the main working on a research project to understand chromosomes. The yeast cells are unable to synthesize adenine and leucine. You have a double octocurve. They can't make their own adenine, they can't make their own leucine. The plasmid that she's working with consists of origin of replication, so okay, cool, it can replicate, and the ADP gene. Following perpetuation of her plasmid into her yeast, she will type of this feels so good. So we play it herself and select herself that picks up the plasmid. What are all the components necessary for this selected media? Select all that apply. So the two answers that she needs: media containing leucine and media lacking adenine. And the question is why? So we have a double octocurve. I need adenine, I need leucine in order to live. I replace one, I give it the gene for adenine, so it doesn't need adenine. And that's why you take media lacking adenine. That kills off other stuff. But then I also need to have leucine because I haven't replaced my inability to make leucine. Right there?
So we're using not affected. Just not.
Right, it just doesn't make leucine. Yeah.
And is this asking like, will it go or is it asking that
It's both. It's what does it need in order to work? I said you transform, the thing that works, I said it has organ application, you know, some capacity is going to work. And it's asking what media do I need for it to actually work? Okay, any other questions? Yes.
So it's the same like question that
Okay, cool.
So obviously it doesn't have organization, but if there wasn't any something.
Yes. So no organ replication, no replication, no colonies. No centromere, small colony. Telomeres is not going to affect the amount of colonies. Only time you need a telomere is if your plasmid is linear. Remember, telomeres protect linear DNA, and we'll actually talk about that lecture on Thursday.
And you have severe conflicts and again, you get covered here.
No, I'm 99.9999999% sure we've covered every topic that's on there. Yes,
But is it possible to get five years
yeah, it was rough, like normal. Yes, here.
I know that secretary and secretary for, like, it doesn't need to open this secretary, and I know how to do this, and even say that the news are, where papers and things. Like, the secretary,
Yeah.
how the secretary, but
If
like,
you-
why can't we do this in the-
Right. If you check the questions down, you'll see that the central questions are all about yeast. The yeast is eukaryote. So it's, yeah, there's the plasmid is circular, but it still needs, like, when it replicates, it needs that centromere or circulate properly. And bacteria, even though they don't have a centromere, they do have a mechanism that allows them to segregate the circular DNA into the same office. So the concept is still true for bacteria, they just don't technically have a centromere. The first question, yeah. Yeah, so it's like I'm doing it, yeah. If you cut it up, then the plasma is kind of useless. I mean, it'll be linear pieces, but it's also a bunch of little pieces of DNA. So it depends. So other times we'll actually talk about like building plasmids, I think in lecture 7 and 8. And then so depending on where the restriction enzyme sites are, sometimes you cut in one place, sometimes you cut in multiple places. And usually we try to design a plasmid to have one site so that you linearize it. And then usually clone something back into it and put it back into a circle. You don't leave it as linear. If you're gonna leave it as linear, it needs to.
Yeah. I want to talk about, the origin of the business, the project. But they really don't think about it.
Correct.
So, in that situation, I think you use the thinking that you're being transmitted, have you knocked out the everything from the system, not going to happen? Or the part of it is not happening.
And so if I tell you there's no origin of replication, just assume there's not one. The way that can come about is many ways. It could be like scientist error. The order of a plasmid that was basically for bacteria wanted to use it in yeast, so it needed origin of replication, and they'd start with one. Depending on the build of the plasmid, it just may or may not have one. And being cognizant of what you need it for allows you to add it or clone it in. Sometimes you can accidentally clone something out because you use the wrong restriction enzyme. Like, oh crap, cut out origin of replication. Like that can happen too. So just know that when I tell you there's no origin of replication, you don't have to worry about why there's no origin of replication. Just know it's not. Here.
you see very linear, like, some periodic.
Actually, I don't remember. Does anyone work with yeast? Is yeast DNA linear or circular? I'm pretty sure it's linear. Right? That's where we discovered telomere. It's linear. It's linear and they can propagate circular plasma. Okay. Yeah, how excited, like digging my memory. It's like, I know this. It's still good. Yeast, okay. Other questions? Yeah. Okay, so we're going to go over this again for why, if there's no centromeres, there are small colonies. So think about cell division. Basically, you have one cell, and then it will have its plasmid, for example, the centromere, and it replicates the plasmid, so there's two of it, and it's supposed to divide and make two cells. But what can happen is that it can start dividing, this piece blends off, and you get a cell that has none of the plasmid. So then the cell without the plasmid is gonna die because you can get the selection of resistance markers. And then so instead of every single time you get two cells upon division that will live, sometimes you only get one cell that will live, meaning you have a slower rate of growth and that you have a smaller complex. Okay, other questions? Yeah.
So your country, or kind of doesn't have. We want to present that.
You use the cell for a plasma that you're getting. The yeast itself wouldn't be able to live without it, right? Yeah. Incorporate telomere into what, plasmid, like a. Yeah, so you could put a plasmid that basically, okay, so let's start with the cell. It doesn't have telomeres, and then the question is whether or not it basically has telomerase, so it can like extend into the DNA. And so if you built a plasmid that could basically like recombine into the actual host genome that has a telomere sequence already, then you use telomerase and extend it, to kind of like rescue the cell. That's super theoretical. Okay. But yeah, technically, yes.
This is not really, and not on my assumption, but for Houston too, and then you said they're like only modified and there's like methylation and phosphorylation.
Yes.
But like, what does that mean? Like, what's the difference in tails?
Okay, so the thing I want you to know about histone tail, and hopefully you learn more in your upper div classes, that the histone tails are what's important for histone to histone interaction. So if I want two histones to compact together, you need the histone tails. They allow compaction of the DNA to make it like 30 nanometer fiber. That's what histone tails are for. And then the whole idea is that these histone tails can be modified. These modifications will change how they interact with each other, basically opening chromatin and closing chromatin. But you only know the details of like which modifications cause what direction of opening or closing. Yes.
Something,
Mhm.
it has something, but not to get for like it's non.
Centromeres gives you sufficient growth, so large colonies. A lot of growth is when there's no selection. So that means everything you put on the plate can grow. So two separate concepts. Yep, if you had complete media, no antibiotics, that would cause a lawn to grow. Yes. So this is a hypothetical. I'm going to start off. What do we know about transformation? Okay, so not 100% efficient. Anything else that we know? So that's selection afterwards. Other thing we know about transformation is that it just kind of happens. And there's multiple conditions and where it happens. Some of it's understood by scientists, some of it is not understood. So when you get the result after a transformation, that there are no colonies. Ignoring transformation, what could go wrong or what could you do that causes no colonies? What are things that cause things not to grow? You have no order of replication. Anything else? You said didn't pick up the plasmid? So didn't pick up the plasmid. No plasmid. You could have the wrong growth conditions, right? So growth conditions. That'd be the media conditions. Yeah, so if I didn't get it, they put antibiotics and everything died. These are all the types of things that would cause you to see no colonies. And then so if it didn't get the plasmid, that means something happened to transformation. We don't know why. Like this happens all the time in the lab. Like go transform, just no colonies. Oh man, something didn't work. You have to go troubleshoot what didn't work. So just know that these are the reasons why you could have no colonies. There could be no order of replication. It could be there's no plasmid because the transformation didn't work. Or it can be because your growth conditions were incorrect and killed everything. Okay, is that clear? Yes. When you have a lawn, yeah, you usually have usually a mix of different types of bacteria. So just to reiterate what was said, colony is when you have a group of bacteria or yeast, whatever that's growing, that's genetically identical. A lawn, you'll have usually a mix of genetic backgrounds. So usually the things that were transformed and things that weren't transformed. Okay. Any other questions? We're at 49. Last chance, last call, then go study hard. Are y'all ready for this?
Yes.
Yeah.
Yeah, that's what I want to hear. There's only 1, 2, 3, 4 people. Okay, I'll see y'all tomorrow.